In this PowerPoint, we are going to focus on the last two learning intentions from page 36 of your student guide. In this PowerPoint, we are going to focus on describing the process of ATP synthesis occurring at the inner mitochondrial membrane, and we're going to state the role of ATP in the transfer of energy. So a reminder from National 5, comparing our learning. We had been looking at the process of glycolysis, and we've looked at the process of the citric acid cycle. What we can focus on at higher is the production of the ATP. At National 5, we just kind of magically talked about ATP being made as a result of enzyme-controlled reactions. At higher, we look at this in more detail in the third stage of respiration, called the electron transport chain. The story we have so far is we've looked at glycolysis and the citric acid cycle. As a result of glycolysis, we have produced two molecules of ATP, but generated hydrogen ions and electrons to be bound to the carrier NADH. In the citric acid cycle, we see further breakdown of intermediate molecules, again generating some ATP, but not a lot. More importantly though, again, the release of these hydrogen ions and electrons, which have been carried by the NADH to this final stage. It's the hydrogen ions and electrons that's been carried by the NADH that is of great value and significance when we look at the generation of ATP in this final stage. The electron transport chain involves using the hydrogen ions and electrons carried on the NADH from glycolysis and the citric acid cycle in the generation of ATP. An electron transport chain consists of a group of carrier protein molecules. There are many of these chains in a cell. They are found attached to the inner membrane of the mitochondria. NADH from glycolysis and the citric acid pathways releases their electrons and passes them to what we call the electron transport chain. It's in this final stage of respiration that we see the most ATP molecules being generated. Already highlighted that an electron transport chain consists of a group of carrier protein molecules. What we have on the diagram in front of you is this full electron transport chain. The inner mitochondrial membrane is represented by the kind of green band. We've got our wee phospholipid molecules there. And the protein molecules are represented by the purple elements on the diagram. So these proteins are forming channels or carriers along that membrane. The first part of our electron transport chain is seeing the hydrogen ions and electrons that have been carried by the NADH being brought towards this inner mitochondrial membrane. So we can see at the bottom left-hand side, it says mitochondrial matrix, and above that we have these NADH molecules which have been produced as a result of glycolysis and the citric acid cycle, carrying with them the hydrogen ions and electrons. In the first instance, we are focusing on the NADH passing over and releasing these electrons and passing them through the protein molecules that appear in the membrane. As the electrons flow along this chain, they release energy. The energy that has been released by the electrons passing along that electron chain is used to pump the hydrogen ions, which was the other thing being carried by the NAD. It's used to pump the hydrogen ions across the membrane from the inner matrix to what we call the intermembrane space, where a higher concentration of hydrogen ions is maintained. So from the diagram, we can see that the hydrogen ions, being represented by the H plus in the blue circle, are now being pushed through these protein channels to that intermembrane space. Now, because of the energy being released by the electrons passing along the protein chain, that energy is able to constantly push the hydrogen ions from the matrix out into the intermembrane space. The return flow of the hydrogen ions back to the matrix is via an enzyme called ATP synthase, which you can find embedded into the membrane, and that's the one that's highlighted on the screen in front of you. The ATP synthase drives the synthesis of ATP from ADP and inorganic phosphate, which is represented down at the bottom right-hand side. Most of the ATP generated by cellular respiration is produced in the mitochondria in this way. When the electrons come to the end of the electron transport chain, they combine with oxygen, the final acceptor. At the same time, the oxygen combines with a pair of hydrogen ions to form water. In the absence of oxygen, the electron transport chains do not operate, and this major source of ATP becomes unavailable to the cell. Let's look at the electron transport chain one final time in one full swoop. An electron transport chain consists of a group of carrier protein molecules represented by the purple elements on the diagram. There are many of these chains within a cell. They are found attached to the inner membrane of the mitochondria. NADH from glycolysis and the citric acid pathways releases electrons and passes them to the electron transport chains, which you can see happening in the bottom left-hand corner. The NADH is passing its electrons, represented by the green circle, to the electron transport chain, which is the purple element. There's that passing over. As the electrons flow along a chain of electron acceptors, they release energy. This energy is used to pump the hydrogen ions, represented by the blue circles, across the membrane from the matrix to the intermembrane space. This is all still within the mitochondria. The hydrogen ions do not leave the mitochondria. This higher concentration of hydrogen ions is constantly maintained because the energy released by the electrons is always pumping the hydrogen ions from the matrix to that high area of concentration. If we go to the far right-hand side of the diagram now, we see the return flow of the hydrogen ions. This is the one where the arrow's pointing down back to the matrix. The return flow of hydrogen ions to the matrix is via a molecule called ATP synthase. This return flow drives this enzyme to synthesize ATP from ADP and PI, which you can see at the very bottom right-hand corner. Most of the ATP generated by cellular respiration is produced in the mitochondria in this way. When the electrons come to the end of the electron transport chain, so you'll see the green dots start to come down and go back to the matrix, These electrons combine with oxygen, which is regarded as what's called the final acceptor. At the same time, the oxygen combines with a pair of hydrogen ions to form water. In the absence of oxygen, the electron transport chains do not operate, and this major source of ATP becomes unavailable to the cell. ATP is important because it acts as the link between catabolic energy releasing reactions such as respiration and anabolic energy consuming reactions such as the synthesis of proteins or the replication of DNA. ATP provides the means by which chemical energy is transferred from one type of reaction to the other in a living cell. It has been estimated that an active cell requires approximately 2 million molecules of ATP per second to satisfy its energy requirements. This is made possible by the fact that a rapid turnover of ATP molecules occurs continuously in a cell. At any given moment, some ATP molecules are undergoing breakdown and releasing the energy needed for cellular processes, while others are being regenerated from ADP and PI using energy released during cell respiration. I would ask you to evaluate your own learning based on the 2 learning intentions shown on the screen. That's us at the end of cellular respiration, so you have your variety of summary and retrieval practice activities available in your student guide. You have your fill in the blanks, which leads to a Level C summary on page 37. On page 38, you have your completed summary again to Level C. On page 39, you have your retrieval practice flashcards. And on page 40, you have your retrieval practice mind map. And via Teams or in class, you have access to these targeted past paper questions on cellular respiration.